Today we're going to talk about skeletal muscle structure. In fact, we'll talk a little bit about all three types of muscle. If you recall, there's skeletal muscle. That's what moves our body, our limbs, and that's what this guy has really beefed up in his body. I think this is photoshopped. No one can get that big. But it emphasizes the skeletal muscle. So the part of your muscle that can move your body through the environment and interact with your environment. Remember, there's also cardiac muscle, and that's the main tissue associated in your heart. So that's contractile tissue that puts pressure on the blood inside of the chambers of your heart so it can provide movement to the blood. Okay? And then there's smooth muscle. And smooth muscle is associated with many of your other organs, such as the digestive system organs, things like your stomach, your esophagus, your small intestines, your large intestines. These all have smooth muscle to help move food and fluids through those systems. Your urinary bladder has smooth muscle. So all different types of organs have smooth muscle associated with them. Your arteries, your veins. Okay, so throughout the semester, we'll be talking about smooth muscle. All of these different types of muscle types actually add up to about half of our body weight. That's how much muscle we have in our body. So it's pretty important. Today, we're going to focus on skeletal muscle, however. And in this lesson, what we'll do is we'll go over the mechanism that makes skeletal muscle contract. Okay, and this will be the sliding filament theory. So some of you may have learned it already in anatomy, and yes, it's coming back. We're going to add a couple of important receptors on there in order to understand the physiology. So we will talk about the sliding filament theory, and then we'll also talk about the neuromuscular junction, how a motor neuron can stimulate skeletal muscle to contract. Okay, and so the items that you learn here will also translate mostly to the cardiac muscle when we study that in the circulatory system, and the smooth muscle when we study that in the various other organ systems that we'll look at. Okay, so first let's look at those three different types of muscle and how we can categorize each one of these. And so if we looked at them under the microscope, the first one that you see there to the left is skeletal muscle. Notice these are long cylindrical cells, but they are just a cell, just like the cell that you learned about in general biology. Okay? They have a cell membrane. They have nuclei. You can see the nuclei are kind of pressed up against the side. They have all the other different types of organelles. They have a lot of mitochondria. But today we're going to learn about other structures inside of there that are very important to allow these cells to contract. Okay? Those other structures form what are called sarcomeres, and that's the stripes that you see within the skeletal muscle there. You can also see the striping within the cardiac muscle. You see it very faint in there. Those are also cylindrical cells, but they're a little shorter than skeletal muscle. Skeletal muscle cells can be very long and span the length of your arm or your upper leg or something like that. Okay, but those stripes in there, we refer to that as striated. Okay, so we say these muscle cells are striated because we can see that those stripings in there. Okay, and so that's one way that we can categorize the three types of muscle. It's either striated, like skeletal muscle and cardiac muscle is, or it's unstriated, like smooth muscle. Smooth muscle does not have this arrangement of the sarcomeres that we're going to look at today. Okay? It has the proteins associated with it, but they just have a different arrangement, so it doesn't show up as striped. Now, another way we can categorize muscle cells is based on how we can control those cells, right? So skeletal muscle, we mainly control by voluntary thought, voluntary movement. Okay? And so that means that the nervous system connection comes from the motor cortex in our cerebrum. So there's a conscious thought into moving our skeletal muscle for the most part. There's always exceptions to the rules in physiology. But in cardiac muscle and smooth muscle, these muscles are mainly controlled from other areas of the brain where there isn't conscious thought. Okay, and so we say that's involuntary muscle, and that's a good thing because you don't have to sit around thinking every 60 to 70 every 60 to thinking each second. You don't have to think 60 to 70 times each minute for your heart to beat. You don't have to will it to do that. It'll do it on its own. Okay, the smooth muscle in your digestive system, it's going to contract on its own. So again, that doesn't involve conscious thought, so that then we can classify it as involuntary muscle. So you've got to remember those two things, whether a muscle cell or type of muscle is striated or not, okay, and then also if it's voluntary or not. Okay, let's look and see the structure of skeletal muscle, how it's put together, and that will also then bring us down to why skeletal muscle is striated. We're going to actually see the structures that make the skeletal muscle striated. So again, some of this should be reviewed from anatomy, and what you see in the top panel there is that a whole skeletal muscle, your muscle like your biceps brachii or something like that, it's surrounded by a connective tissue called epimysium. Okay, so it's just the connective tissue that surrounds your whole muscle tissue. Okay, but if we go down further, you can see within skeletal muscle that there are different little structures in there referred to as fascicles. So skeletal muscle consists of these bundles we refer to as fascicles. Okay, and fascicles are surrounded by a connective tissue called perimysium. Okay, all up and down the length of fascicle is perimysium. Now, within the fascicles themselves, those consist of bundles of muscle fibers. Okay, muscle fibers. And a muscle fiber is just another way of saying muscle cell. Remember that skeletal muscle cells are very long. And so many times we refer to them, instead of saying cells, we say fiber, because it reminded early anatomists of protein fibers or something like that. But we know it's not a protein. We know it's actually a cell. Okay, so just keep that in mind. A muscle fiber means the same thing as a muscle cell. It's very important to remember that, okay, because we can interchange that word, and that does happen often in your textbook and in other readings. Okay, individual muscle fibers then are surrounded by endomysium. Okay, and if you see those terms, epimysium, perimysium, endomysium, hopefully you're thinking that sounds really similar to what we learned with our nerves. We had epineurium, perineurium, and endoneurium. So it's the same type of connective tissue forming these, but just surrounding either nerve cells or muscle cells. And bundles of muscle cells are fascicles, just like we saw with bundles of nerve cells, and so on and so forth. Okay. Okay, so within a muscle fiber now, you can go down to the third panel. Again, within a muscle fiber, a cell, it is a normal cell. So it has mitochondria and nucleus and things like that. But the other structure that is very important to skeletal muscle is the myofibrils. These are proteins that run the length of the entire muscle cell. And these proteins are the functional portion of the skeletal muscle cell. This is what helps the skeletal muscle cell to contract. Okay, so these myofibrils, and we're going to get a little more detail of these proteins, but they're arranged into what are referred to as sarcomeres. And that's actually what you can see at the bottom, the last panel there. But each sarcomere is wrapped around an organelle called the sarcoplasmic reticulum. And that's what you see in the second to the last panel, the sarcoplasmic reticulum. This is a series of membrane that surrounds each myofibril, and its main job is it stores calcium. And so it's going to control the release of calcium over the sarcomere, and also remove calcium that's surrounding the sarcomere at specific points. And so we're going to see that calcium is going to be very important at triggering the contraction within these sarcomeres. Okay. So the myofibrils I mentioned are arranged into sarcomeres. We'll talk about that a little bit more in a minute. But each myofilament, these are made out of proteins called actin and myosin. So we're going to discuss those a little bit more. First, let's look a little more at the cell structure itself. Okay, and so this would be looking inside just a portion of a skeletal muscle cell. A skeletal muscle cell has a cell membrane, but if you've had anatomy, recall that in muscles, instead of calling it the plasma membrane, we call it the sarcolemma. Okay, sarco refers to flesh, meaning muscles. Sarcolemma, that's in bold up there. That's just the cell membrane. So muscle cells are excitable just like neurons, and so a neuron is going to be able to pass an action potential to the sarcolemma of a skeletal muscle cell. So action potential will run down that membrane. You can see then the sarcolemma invaginates into those tubes that you see that fold down into this within the cell. And so that tube is called a T-tubule. The T stands for transverse because it transverses the cell, means it cuts down through the cell. You can just call it a T-tubule. And so an action potential that runs down the sarcolemma will be continuous and run down the T-tubule because it's just an extension of the sarcolemma itself, cell membrane, going down into the cell. And so remember, you have a round cylindrical cell. We want to be able to bring the action potential down into the inside of the cell so that it can interact with that blue structure there, which is the sarcoplasmic reticulum. The SR, or the sarcoplasmic reticulum, stores calcium. So we're going to see that the action potential stimulates the release of the calcium from the SR. And it's going to flow over those red and purple structures that you see there. Those are the thin and thick filaments, and I'm going to explain what those are in a minute. Notice that the sarcoplasmic reticulum, though, is a series of membrane tubules that surround the sarcomere, and on the ends, it's kind of a fattened sac, and that's called a lateral sac. So one T-tubule, bringing an action potential, can stimulate the lateral sac of one SR on one side and an SR on the other side, right, on the left and right sides that you see there, allowing calcium to be released from both of those SRs on that side of the sarcomere. Alrighty? And you might have learned in anatomy that a lateral sac with the T-tubule in the middle and the other lateral sac together, those three structures are just referred to as a triad. Okay, so again, in physiology, very important to understand these terms and this structure, okay, to make so that we can later understand the function that we're going to start to go over. Okay, a little bit more then on the myofibrils themselves, their structure. So again, a myofibril is a contractile element of a muscle fiber. So these are what are actually contracting. And they have, we refer to them as thin and thick filaments. That's the arrangement of the red lines and the purple lines. You can see the purple lines are thicker, so those are the thick filaments, and the red lines are thinner, so those are the thin filaments. And the way those are arranged, they have this regular pattern that forms these stripes inside of the cell. So when you look at it under the microscope, you see these light and dark bands, the stripes. That's why we say skeletal muscle is striated, because of the striping formed by the thin and the thick filaments. So the thick filaments are myosin, which is a protein, and the thin filaments are mainly actin, which is a protein as well. And so I mentioned that they have a regular arrangement pattern, and that's what you see here that are labeled, okay? The dark areas, or the what you mainly see are the purple areas. That's referred to as the A band. Okay, so the A band contains all of the myosin, or the thick filaments within it. That's the A band. Okay, the I band contains the actin and only the actin. Right now, ignore the green squiggly line. Okay, we're going to ignore that for a moment. I band does not contain any myosin in it, the thick filament, only contains the actin. But notice the H band, while it contains all of the myosin, it also has some overlap of actin inside of it. Okay, now the myosin, the purple lines there, are attached to the vertical line down the middle, the M line. That delineates the middle of a sarcomere. Okay, then notice down the middle of the I bands, there are those zigzag lines. Looks like a Z, and those are actually called the Z line or the Z disc. That delineates the ends of the sarcomere. Okay, so you've got the middle, and then you've got the two ends of the sarcomere. The ends are the Z line, the middle is the M line. Okay, within the A band, I mentioned there is an overlap of actin and myosin, so we actually refer to that as the zone of overlap. And again, that's also labeled within here. So what's going to happen to get a contraction? The actin and myosin are going to interact with one another. The myosin will actually be able to link with the actin above and below it, and it's going to pull the actin toward the M line. And actin is attached to those Z discs. So when actin is getting pulled toward the center, it's going to pull on those Z discs, and the whole sarcomere is going to shorten. Okay? The whole sarcomere is going to shorten. So actin and myosin don't shorten. Okay, just the sarcomere. So if I put my fingers together like this and you see my thumbs, pretend my thumbs are the Z discs. And my fingers are the interaction of actin and myosin. My fingers are going to slide across one another, and you can see my thumbs get closer together. So I just produced a contraction. But my fingers didn't shorten, they didn't change length. Okay? That's the same thing that happens with actin and myosin. Myosin is going to pull on actin toward the center. The length of actin and myosin will stay the same, but the Z discs will come closer together. Okay, so keep that in mind as we continue through this process. So in order to really understand how that works, now we have to look more deeply at what makes up actin, or the thin filaments, and what makes up the thick filaments, or the myosin. Okay, so let's start with the thin filaments. The thin filaments are made out of three main substances. The main substance are the actin molecules. Okay, and that looks like that string of pearls in the photo indicated as B there. It looks like the string of pearls there. They're orange with a yellow end. Okay, those are the actin molecules, and specifically each individual one is called a G-actin molecule. It's okay if you just call it actin. Notice on it, it has what's called an active site. An active site is where something binds to that area. It allows an interaction with another molecule in that area. Okay? But we're going to control that interaction with another molecule, and we control it by the rope-like structure called tropomyosin. Okay, tropomyosin. You can see that boxed out for you there. Looks like that bluish-green rope that spans the length of the actin molecules, and it's covering the active sites. So as long as that's covering the active sites, there can't be any interaction with another molecule. And what holds the tropomyosin in place are those small brownish rounded molecules called troponin. Okay, so troponin is going to hold tropomyosin in place. Troponin is going to be able, though, to shift position, and when it does, it's going to pull the tropomyosin off of those active sites, exposing those active sites, and that means we're going to be able to get an interaction. Okay, and then notice right underneath the sentence of troponin holds tropomyosin in position, that picture there is showing how actin is attached to the Z line. Okay, so those are attached to the Z line. So if actin gets pulled toward the center, toward the M line, then those Z lines are going to get closer together, much like my thumbs got closer together in that demonstration. Okay, so those are the thin filaments. Now let's look at the thick filaments. The thick filaments are simply myosin, but it's bundles of myosin. A single myosin, you can see toward the bottom there, indicated by D. It looks like a club. Myosin is a long protein. It has a tail. Okay, and then there's a hinge that allows the head at the other end to swivel or pivot. Okay, so that's the whole structure, that one kind of club-like structure there. That's a molecule of myosin. But notice and see that those molecules of myosin all bundle up together. Okay, and that's what's forming the thick filaments. And those are attached to one another via the M line. And again, the M line runs down the center of a sarcomere. Okay, now remember that the A band consists of all of the myosin. So everywhere that you see myosin, that is the A band. But within the A band, you can see indicated on this picture, is the H band. And that is a zone where only myosin occurs. Remember, the A band can also have overlapping section with actin. You can see that there. But there's a portion right around the middle of the M line where only myosin occurs. And that is relatively wide when your muscle cell is relaxed. Because again, with a contraction, if we pull the actin toward the M line, we're going to get more overlap between the actin and myosin. So the H band is actually going to get shorter. Okay. The last structure I do want to talk about, and it's written for you here, that green squiggly line. Okay, that's called titin. And here it is on another slide. So titin, I circled it for you down at the bottom right corner. Titin is a protein that spans the length of the entire cell. It's highly elastic. We believe it's the largest protein in the body, and it extends in both directions from N line to the Z lines at opposite end of the sarcomeres, and it acts like a spring. You can see it's coiled up. So when the muscle contracts, it coils up even more like a spring, like if you're compressing on each end of a spring. So that when the muscle contraction relaxes, it helps the sarcomere spring back to its original position. Okay, goes doing and springs right back. Alrighty, so again, to summarize a little bit, actin and myosin are considered the contractile proteins, although they don't actually contract, meaning they don't physically get shorter. They just pull along one another. Myosin pulls on actin so that the sarcomere gets shorter. The sarcomere is what contracts. Okay, and actin and myosin actually isn't unique to muscle cells, but they're actually more abundant and more highly organized within our skeletal muscle cells. We'll see a similar arrangement in cardiac muscle. That's why cardiac muscle is also striated. Okay. All right. Let's go a little bit deeper now and look at a molecule of myosin. Okay. And so looking at its structure, so now we can understand how it interacts with actin. Okay. So again, it looks like a club, like a golf club. And the part that interacts with actin is the head. Okay, and so you can see there's actually a double head on them. And when the head can reach up and bind with actin, we call that a cross bridge. So when we say myosin is making a cross bridge, that means it's attaching to actin molecules at that active site on actin. So if you look at the upper head on the myosin molecule there, you'll see there is a portion of the molecule that's referred to as the actin binding site. That's the portion that can make the cross bridge with actin, with the active site of actin. Okay? In order to do that, myosin needs energy to do that, and also energy to be able to pull. So it needs energy to make the cross bridge, and it needs energy to pull on actin. And it pulls because it swivels its head. Remember I mentioned on the previous slide, 2 previous slides ago, it's got that hinge. And you can see then the myosin head can swivel. So it can do that. It needs ATP to do that. So that means our myosin head also has an ATP binding site. And there you can see myosin ATPase. ATPase is an enzyme which is going to help split ATP. Remember, ATP is adenosine triphosphate 3. Let me see my 3 fingers in there. Okay. And so if we can break off one of the phosphate groups, then that's where we're going to obtain energy from, allowing the myosin head then to be able to pull the actin. Okay. Alrighty, so you got to remember the actin binding site and the myosin ATP site. All right, let's look at actin. Actin, remember, looks like a string of pearls. This picture is showing actin as blue with green active sites. So those active sites are also called a binding site. That's where the myosin head can attach to and interact with. That's where the cross bridge gets formed. And I've mentioned this already. In order to control, if those active sites are exposed and there's ATP available, then myosin will make a cross bridge and we will get a contraction. So in order to control that, because you don't want your skeletal muscles to constantly be contracting all the time, we control that with that long thread-like molecule called tropomyosin. And here it looks like the brown kind of rope or thread-like. And at relaxation, you can see it lies over the actin molecules right over the active site. So that even if myosin is there with ATP, it can't interact with the active site at this point. So how do we move tropomyosin? We use the other molecule called troponin. Troponin are stationed along the length of tropomyosin. It's about every 6 actin molecule. Okay? And troponin can move. And when it moves, it pulls tropomyosin off the active sites. And if it moves back, it puts tropomyosin back over the active sites. So we see that there's going to have to be a way for troponin to be able to move to pull tropomyosin off when we want it to happen. Okay? And the way that's going to happen is if calcium is available. So now we're going to bring in a molecule of calcium. So the upper panel shows you everything is at rest. The tropomyosin is laying over the active sites. Troponin is there, okay, holding it in place. And you can see there's a myosin head, but it's not forming a cross bridge. Okay, in the bottom panel, calcium shows up. Those little purple balls indicate calcium. And when calcium kind of floods around the sarcomere, remember this is all within the cytoplasm in the intracellular fluid. So if calcium gets into the intracellular fluid, it is attracted to troponin and binds to it. And when it does, remember these are molecules, so the molecule of troponin just kind of changes its shape. And when it changes its shape interacting with calcium, it pulls on tropomyosin, exposing those active sites. Now myosin can make a cross bridge. Okay, but again, there's going to have to be some specific things that occur with myosin. We mentioned ATP for that to happen. So we're going to step through this now to see how put all of these parts together. Okay, Okay, I kind of had to go through and show you the structure of everything. Now we're going to go step by step and see how all of this gets put together. So it's these steps that you're going to need to remember, to need to explain how a skeletal muscle contraction occurs. Okay? And remember, we start that by stimulating the skeletal muscle with a motor neuron. Okay, so here's our motor neuron. Should originate in the central nervous system. Okay, it sends an action potential down to its axon to the synapse, and we call that a neuromuscular synapse. Okay, and you can see that the synapses, the synaptic terminals of our motor neuron, just kind of lie over the top of the sarcolemma of a muscle fiber. So we're just showing you one motor neuron and one muscle fiber. That motor neuron can also branch and stimulate other muscle fibers as well. Let's zoom in on that. Now we can see a little bit more closely the neuromuscular junction. That's the synapse between our motor neuron and our skeletal muscle cell. So at the very close-up detailed view at the bottom there, you can see that the neurotransmitter that is used to stimulate a muscle cell is ACH, acetylcholine. It's always going to be acetylcholine. So it's released from the end of the axon. It then binds to the ACH receptors on the sarcolemma. This is always going to be excitatory, so those receptors are called nicotinic receptors. Okay? They're always excitatory, so therefore those ACH receptors are nicotinic receptors. Remember your subtypes. Okay? Now, the portion of the sarcolemma that is on the other side of the synaptic cleft, sometimes we call that the motor end plate. Okay, that just means that's where the receptors for ACH are found. Okay, and under the microscope, it looks like a stiffened area. That's why it's called a motor end plate. Okay, so all that refers to is the receptor area of the synapse for the muscle cell. Now there's another molecule in there. It looks like a little moon. It's called ACHE. That's an enzyme. We're going to come back to that in a minute. So just know that there's an enzyme that just kind of floats around in the interstitial fluid of the synaptic cleft. It's just always there. And that we'll come back to at the end of our steps. Okay, so our action potential gets generated on the sarcolemma. Okay, and that AP, several action potentials travel down the sarcolemma. And remember that the sarcolemma, the cell membrane, is continuous with the T-tubules. So the AP travels down the T-tubules. And those T-tubules are associated with the lateral sacs of the SR. So it stimulates the SR to release calcium that's stored inside of it. That's where our calcium comes from. And remember, the SR is surrounding each sarcomere, and the sarcomeres run along the length of the entire skeletal muscle cell. So there are several sarcomeres, and each one has their own SR with calcium in it. So calcium gets into the cytoplasm. And once it does that, here we see that here, it binds to the troponin. Alrighty, and the upper picture shows you troponin not without calcium. The bottom picture shows you calcium now is in the intracellular fluid, and it's bound to troponin. Troponin then changes its shape, and it pulls tropomyosin off the active sites. We call that a rotation of the troponin-tropomyosin complex. Our active sites are exposed. Okay, so now we can see what happens, what's going on with myosin. So this is what's going on with the actin portion once calcium gets released. Now let's see what's happening with myosin so that we can get a cross bridge here. Okay, here's our myosin. So remember, myosin needs ATP. And remember that myosin has an ATP binding site. So ATP binds there. Our skeletal muscles are full of mitochondria, just chock full of mitochondria. There's a lot of them in there, and they're making ATP all the time. So there's ATP swimming around in the cytoplasm. Okay, so it binds to the myosin head. Okay, and then remember I mentioned that's ATP binding site. We said it was an ATPase. So it's actually an enzyme that can hydrolyze the ATP. Hydrolyzing means it uses water to break off one of those inorganic phosphates. And that's what you see over to the right. Now the myosin still has ADP and the inorganic phosphate bound to it. Okay, so it's not ATP anymore because the inorganic phosphate, we broke that bond. That released energy to allow the myosin head to straighten up, and lift up and bind to the exposed active site. Okay, so these things are happening at the same time. The actin has to have calcium bind to the troponin so that the active sites on actin are exposed, and myosin has to have an ATP bound to it. And when it hydrolyzes, that myosin head is going to move upward, right? So that's a movement. It needed energy to do that. And now we have a cross bridge. Okay? But now the myosin head has to pull. Okay? It has to pivot its head to pull. Well, how does it do that? It does that when the inorganic phosphate gets released. You can see the little white ball there pops off. That release also releases energy, causing the myosin head to pivot, and it pulls on the actin molecule. Okay, so we're showing you one myosin head doing this. You've got to imagine, remember there's a bundle of myosin heads. They're all doing this at different times and pulling. And they're pulling each side of the sarcomere toward the center. Right? And so our actin then is going to pull on the Z discs, and our sarcomere is going to get shorter. Okay, so let me show you this action of the myosin head kind of in a circle. Okay, so if we start at the top there, or if we start at the over here on the left, you can see our myosin has ATP bound to it. We're going to assume now we have calcium up on the troponin. It moved the tropomyosin, and the active sites are exposed on the actin. So ATP hydrolyzes, means it breaks up into ADP and inorganic phosphate. That causes energy for the myosin head to lift up and form a cross bridge with actin. Then you see the inorganic phosphate being released. It's popping off. That causes the myosin head to swivel and pull on actin toward the M line. Okay. Then the ADP releases too, and myosin head is stuck. It's stuck in that position until a new ATP binds to it. So that moment where the myosin head is stuck and it doesn't have any ATP bound to it at all, that's called the rigor state. It happens in every contraction cycle of a muscle, and it's normal, rigor state. Okay, it sounds like rigor mortis because it's related. When you die, your muscles become stiff, and that's because you run out of ATP, you're dead, you're not making it anymore, and all of your actin heads or myosin heads are stuck. And so there's a brief period of time where your muscles are stiff because the myosin heads are all stuck in a cross bridge. Okay, but then the tissues break down and it releases over time. So rigor state is a normal state that occurs during every contraction cycle, okay, while you're alive. It's a brief moment where there's no ATP bound to myosin. it's different than rigor mortis, but it's related because when you're dead, you don't have any more ATP. And so after death, for a period after death, all your cross bridges are stuck until tissues can break down. All right, so it's important to distinguish the rigor state. Okay. All right, so we refer to this as excitation contraction coupling. Okay, because our motor neuron is what's stimulating the release of calcium. You cannot get a contraction unless calcium interacts with troponin. Okay, and so the AP coming from the motor neuron is transferred from that motor neuron to the sarcolemma, down the T-tubules, to the sarcoplasmic reticula. Okay, so again, we're showing you one muscle fiber here, or a muscle cell, and on the inside, we're referring to one myofibril. But if one muscle fiber has an action potential, that action potential just spans across the entire muscle fiber. And there are just several T-tubules all along there, interacting with all the different sarcoplasmic reticula inside the cell. So there are hundreds and hundreds and hundreds of them inside the cell, all associated, each with those individual myofibrils. So each of those individual myofibrils contain hundreds and hundreds of sarcomeres laying end to end with one another, and they're all contracting. Okay, so this shows you a little bit of a cutoff section. You see one sarcomere in front of you. Now the sarcoplasmic reticula is green, but it's cut off on the left side and it's cut off on the right side. So really we're seeing three sarcomeres. Okay, but they're cut off on the left and the right side. Okay, so just remember the SR, sarcoplasmic reticulum, is a modified endoplasmic reticulum. So you learned about those in general bio, but here their job is to store calcium. Okay. And so you can see the T-tubules, which are connected to the sarcolemma, they invaginate, and then they can span the entire length of the cell, or the diameter of the cell, reaching all of the different myofibrils within there. And each one of those have their own SR. Okay. So all of those, that action potential is going to zip down the T-tubules, and all of those SRs are going to release calcium. Okay. All right. Almost done here. We got to keep going though, because now we have to see, how does the action potential release calcium? So we're going to look at voltage-gated channels associated with a calcium channel. Okay, and so that's what you can see down here in the lower right corner. You notice the T-tubule invaginating inward. There's kind of a 3 right in the middle of it. The bluish sacs on either side, those are the lateral sacs of the SR on either side. And there you can see the receptors that are associated. And so we have 2 that we need to deal with. One is a voltage-gated channel. It's called a VGDH channel. Okay, VG, that means voltage-gated, so that means the action potential is going to stimulate that. Those are the little purple guys there. Those are each associated with a channel located on the SR. So the voltage-gated DH channels are located on the T-tubules. The calcium channels are located on the SR. Those are also known as foot proteins. So different sources, it'll call it a calcium channel, or it might call it a foot protein. So you want to be aware of all of those. So let's get a detailed view of the voltage-gated channels and the foot proteins. That's what we see here. So the green is indicating a portion of the SR where the foot proteins are located. Again, those are calcium release channels. So when those are stimulated to open, calcium is going to flow out of the SR into the cytoplasm. It's going with its concentration gradient. There's a lot of calcium in the SR, not so much in the cytoplasm. Okay, but what stimulates those calcium release channels or foot proteins to open are the DH receptors, the voltage-gated DH receptors. DH stands for dihydropyridine receptors. You can just remember DH. But you also must remember it's voltage-gated. Those are located on the T-tubules. So that makes sense. If the sarcolemma is continuous with the T-tubules, so the action potential comes down the sarcolemma, down the T-tubule, and the action potential then stimulates the voltage-gated DH receptors, okay, to then activate and open the calcium release channels or the foot proteins on the SR. And the end result at the one on the right, you see calcium being released. Now, calcium is going to associate with our troponin. Troponin then will move, pulling tropomyosin off of the active sites of actin. At the same time, myosin heads have an ATP bound. It hydrolyzes. That gives it energy. Now the active sites on actin are exposed, so the myosin has energy to make a cross bridge. The inorganic phosphate pops off. That gives energy for the myosin head to pivot, pulling actin toward the M line. Okay? And it wants to do it again and again and again. So it releases the leftover ADP. It's stuck in rigor. A new ATP binds. When that happens, the myosin head can release. Then it hydrolyzes, get another cross bridge. Inorganic phosphate flies off. We get a pulling. The ADP releases, it's stuck. A new ATP, it releases. And it just keeps doing that as long as there's ATP and as long as there's calcium. Okay, and that keeps happening. There's always calcium as long as the motor neuron is firing. So how do we get it to stop? Well, that's the relaxation of the muscle. First thing that has to happen is your motor neuron stops firing. Okay? And we need then to clear the calcium out of the cytoplasm. So we do that by first, again, the motor neuron stops firing because we have to stop the action potential. As long as there's ACH in the synaptic cleft, it's going to bind to the receptors, and we're going to get the AP on the sarcolemma and down the T-tubule, and we're going to continue to stimulate calcium release out of the SR. So remember, I mentioned those little moon-shaped pictures, moon-shaped molecules in the previous picture. I said that was an enzyme called ACHE. It's called acetylcholinesterase. Remember, ACE means enzyme. ASE means enzyme. So this is an enzyme that breaks down ACH. So that's how we clean the ACH out of the synaptic cleft. ACHE, it's always in there, and it just starts breaking down acetylcholine. And when the motor neuron stops releasing acetylcholine, then the ACH can catch up and clean it out of there. Now our receptors aren't stimulated anymore, so there's no more action potential. So the SR is not releasing calcium anymore. But we also have calcium pumps. And so ATP is running calcium pumps to draw the calcium out of the cytoplasm and put it back into the SR. That means the calcium is no longer bound to troponin, so our tropomyosin can cover the active sites on actin and no more cross-bridging can occur. And when that happens, you can continue the pictures down below the myosin filament. You can see our sarcomeres will go back to their regular length and your muscle lengthens and back into its relaxed state. Okay, so I've got some videos on Canvas to show this action, and it steps you through everything. So you will have to know the steps of the slide. This is called the sliding filament theory, or how a muscle actually contracts. Okay, so you want to start with the stimulation of that motor neuron, and then all of those steps that happen after that. And I've gone through it several times in this video. Okay, and it'll in the check your understandings, I have you write that out. You want to list it out. Alrighty? Make sure you can do that. Now, I want to end this with just talking about ATP because we talked a lot about the mitochondria earlier and how important it is for that organelle to make ATP. And it's really important in muscle cells because we use ATP in a lot of different areas, right? So ATP is needed to bind on the binding site of myosin so that it can cross bridge and that it can also do the power stroke. I may not have mentioned that before, but that term, the power stroke, means when myosin is pulling on actin. Okay, so ATP is needed for those actions there. Okay, it's also what causes the rigor state. The rigor state occurs until a new ATP binds. Okay, so that's how we get continuous power strokes or successive power strokes. And that's what helps to keep the muscle contracted. Okay, then it's also needed in relaxation because we need the ATP to reuptake the calcium. Okay, we don't need it between the actin and myosin. Relaxation just occurs when calcium's gone and myosin is no longer making cross bridges because tropomyosin is covering the active sites. But in order to get tropomyosin to cover the active sites, we have to remove it from troponin, and that happens when ATP is powering the calcium pumps that pump calcium back into the SR. Okay, so that's it. That's the sliding filament theory. That was a long one. I'm sorry that was long, but we had to do it all in a continuous process. Okay, make sure you check out the videos on Canvas and study your sliding filament theory. Hopefully this is the second time because you learned it in anatomy.